Hi guys, let's start talking about lab number 2, ubiquitous microbes. This is one of my favorite labs to do when we're face to face on campus because it gives you a stunning realization of just how prevalent bacteria are, that they are everywhere. Ubiquity just means it's everywhere. Uh, so this lab was supposed to demonstrate that. They are found everywhere. They're in air, dirt, water. They're in plants. They're in the mountains. They're in the hot springs. They're in the at the South Pole. And a common characteristic, of course, these things are too small for us to see. So the kind of organisms we're referring to are the bacteria, the archaea, protozoas, algae, fungi, viruses, viroids, and prions. We're in microbiology, we're going to talk about how these guys react with our immune system, positive and negative. Um, Although a lot of these bacteria are doing more than simply being disease causing, some of them actually help us in a lot of positive ways that we actually would have a hard time living without them. These guys, I'm not going to repeat everything that's in this introduction because quite a lot of it, guys, to be honest with you, is the same as that we have in the lecture PowerPoint. I'll let you guys read this. Um, some things, though, I do want to make a mention on. Some bacteria are specific, usually for certain environments. There are some bacteria that grow best at room temperature, which is around 75 something, 72 degrees Fahrenheit. And then we have other bacteria that grow best at human body temperature. It makes sense to me, and hopefully to you too, that the ones that are most likely to be disease causing, although not absolutely, but the disease causing guys are mostly going to be in that category because that's they are so constituted as if they will have the maximum growth and efficiency while they are in contact with temperatures close to our body temperatures. Sorry guys, trying to straighten this thing up. Okay, oops, made it worse. And so these guys don't grow as well on, say, tabletops or door handles, things that are lower. They also don't grow very well if there's something hotter, and that's kind of the situation. There we go. We got that thing straightened out. So this is a consideration. Now also, there are bacteria who have the need for oxygen, just like we do. There are others for whom they can live in an oxygen atmosphere, or they can live in an anoxic, an anaerobic, a no oxygen atmosphere. And there are some that absolutely require a no oxygen atmosphere because it's actually toxic to them. This is probably the main point I want you to see. There is no one condition under which all bacteria will grow, or microorganisms. They each have their own specific needs. Just like you wouldn't expect to see a polar bear at the equator, nor would you expect to see a tropical fish at the North Pole. Every form of life has its own particular set of environmental conditions that are optimal. So the same thing is going to be true for bacteria. In this lab, one of the things that we were going to do was we were going to actually take a swab and swab things like cell phones and door handles, stair rails, and see the type of bacteria that we find on there and be very surprising. One of the things that surprises everyone is that usually on the campus they swab the bathrooms and they are much cleaner because they receive extra effort, so they generally stay a lot cleaner than other places. Okay, let's see what else we can say in here, guys. Sample materials and from the body. We we generally get that we have folks wash their hands and then we take bacteria off their hands and see what grows there. And then when we take these samples, we're going to put them in little oven-like things called incubators. One of the incubators we're going to set for room temperature, and the other we're going to set for body temperature and grow those. We grow these guys by putting them on this little circular plate, petri dish, and inside that is a kind of a gel-like mixture called agar. That agar generally has nutrients in it that will help the bacteria to grow. Because if we're going to grow bacteria, and we talked about why we wanted to grow bacteria, then you obviously have to provide them with not only the heat that they need, either the right temperature, but we also have to provide them with the food that they're going to need. The agar, we actually pour it as a liquid and it sets and gels up. Trypticase, soy, agar are just the TSA plates. Those are the normal ones we use. We use a test tube of sterile water because we don't want to find out what bacteria are in the water. We want to find out what was on the door handle or on your hand or whatever. So we get those. We write 25 on one, which is room temperature in Celsius, 37 on the other, which is body temperature in Celsius. When we do that, we always label those plates on the bottom because these guys have lids, tops, but lids can come off. The only place you can actually write it is the bottom. And in order to be able to see the bottom, we actually write it on the bottom, around the edge at the bottom in a kind of a circle. We'd put something like the date, the time, where we got it from, things like that. And also, since we have multiple plates being there from multiple different groups, we actually put some sort of identification feature. Because you come back 2 days or a week later and you're not going to remember whose is whose. Okay, we already talked about that note. So what they're doing is getting the sterile water and swab the hands, and then they're going to put it on a plate, and then they're going to have the person wash their hands and do it again. And you'll be surprised that how much bacteria are left after we wash our hands. And then generally we get they're saying 4 to 5 TSA plates and have different members of the group swab different things. Shoes, cell phones, whatever. Money is one of the dirtiest things that you will actually come across. It is very germy. Okay. Okay, when we came back, we were going to look at the bacteria, the growth on there. Now, when we see these colonies, they're going to be kind of circular, but the exact formation of the colony is something we're going to note. Why? Because different bacteria form different colony shapes. Punctiform, circular, filamentous, irregular, rhizoid, all of these. Okay, so we want to look around and see in general what the shape of the colony is. And then we're going to look like from the side and see, is this thing pretty flat? Is it a little bit high up? Does it actually have a convex or form an even larger? Or does it form a convex with like a tip on it like this? And then the other thing for colony morphology that we're looking for is the actual border of the colony. Is it entire? Is it unbroken? Does it undulate? Does it make these very deep chasms? Is it erose, filamentous? and that's also a form. Does this thing have little growths coming in, or is it curled like this? Okay, guys. This video, when I tried it, was not available, but this one is. So check this one out. You could try checking that. It may be temporarily offline. I don't know. I'll try to do some research on that. And again, I want you to do these questions, please. Ampled. I think that should be sampled. Yep, I think it's too far apart. There we go. Different surfaces. Okay. Getting our grammar right so I can get these annoying things off there. Well, guys, that's it for Lab 2. Again, since that one video, that's right. Since that one video is not available, please, 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 if there's anything you don't understand about this, please ask me questions, okay? But there are some things, I would especially, all the introductions on these labs contain information. Some of that information is some that you don't hear in the lecture. This is extremely rich fields for mining questions on exams or quizzes rather. Okay guys, that's got it for lab 2.